Dear students of class 12, आज आपका टॉपिक है इनफर्टिलिटी एंड ARTs. Infertility and ARTs. ART means assisted reproductive technology. ART means assisted reproductive technology. ARTs means assisted reproductive technologies. Infertility means the problem lies with the female. When the female is not able to conceive due to any problem, it is known as infertility, or we can say inability of female to conceive. The problem may lie with the female as the female cannot ovulate, or there may be problem in the reproductive tract, uterine problem, fallopian tube problem, फर्टिलाइजेशन के लिए प्रॉपर साइट नहीं होगी और इम्प्लांटेशन के लिए प्रॉपर साइट नहीं होगी. With the result, the female cannot conceive. It is known as infertility. Inability of females to conceive is known as infertility. Now, such females, when they are not able to produce children, they can be assisted by means of some techniques, and these techniques are called ARTs, or we can say assisted reproductive technologies. Two types of techniques are involved to help the females. One is in vitro fertilization and another is in vivo fertilization. In vitro fertilization and another is in vivo fertilization. In vitro fertilization means fertilization is done under the laboratory conditions outside the body of the female, and in vivo fertilization means fertilization inside the body of the female by means of transfer of gamete. In vivo means inside the body of the female. In vitro means outside the body. Outside the body means in vitro. In vitro under laboratory conditions. Now there is a technique of in vitro fertilization which is known as ZIFT. Z-I-F-T. ZIFT. By ZIFT we mean zygote intra-fallopian transfer. Zygote intra-fallopian transfer. Here, zygote is transferred in the fallopian tube, or zygote can be transferred in the uterus if it is beyond 8 cell stage. 8 cell stage से आगे अगर zygote होगा, then it can be called early embryo. This early embryo can be transferred in the uterus. In both the cases it is known as ZIFT, zygote intra-fallopian transfer, but the zygote is formed under laboratory conditions when both female and male donors, they donate the gametes. Female can donate the egg or ova, male can donate the sperm, and these sperms and eggs are taken under laboratory conditions, and the egg is fertilized in the laboratory by means of sperm. When it is fertilized in the laboratory by means of sperm, zygote is formed, and this zygote is then transferred in the fallopian tube of the female, or if it proceeds beyond the 8 cell stage. Beyond 8 cell stage, it can be transferred in the uterus. In both the cases, the further development of the embryo occurs inside the body of the female, and a normal child is born after implantation, after further development, and the female couple is assisted. Female as well as the male, both the couple get benefit by means of this technique, which is known as ZIP. In vivo fertilization, in vivo, there is a technique which is known as gift, G-I-F-T, gift. By means of gift, by the term gift we mean gamete intrafallopian transfer. Gamete intrafallopian transfer. Gamete intrafallopian transfer. Gametes are transferred, not zygote, not early embryo, but the gamete is transferred into the fallopian tube. fallopian tube में gamete transfer किया जाता है. That is, the donor female and the donor male, they donate the gametes, and the gametes are transferred into the fallopian tube. Egg as well as sperm, these are the gametes, they are transferred into the fallopian tube, where fertilization occurs normally. Fertilization occurs in the fallopian tube. Zygote is formed in the fallopian tube, and further development occurs in the female reproductive tract for the formation of the fetus, and a normal child is born. But it is in vivo fertilization. Fertilization occurs inside the body, not under the laboratory conditions. There is another technique which is known as intracytoplasmic sperm injection. Intracytoplasmic sperm injection. Sperm is injected into the egg. Egg में sperm inject किया जाता है by means of a syringe under laboratory conditions. It also occurs under laboratory conditions, which is known as intracytoplasmic sperm injection. The egg is injected a sperm fertilizes the egg. Again, the zygote is formed in the laboratory, and this zygote can be then transferred, but the egg is fertilized by means of the injection. That is why it is called intracytoplasmic sperm injection. By means of injection, sperm is introduced into the egg. It is again in vitro fertilization. Now, there are artificial insemination techniques, AITs. AITs means artificial insemination techniques. Artificial insemination technique. In artificial insemination technique, the sperms are introduced into the vagina or into the uterus of the female genital tract, and then after insemination, the egg gets fertilized normally inside the body of the female, but the semen is artificially introduced into the body of the female, and further development occurs in the body of the female till the birth of the child. It is known as artificial insemination technique. These were various assisted reproductive technologies to assist the couple for the formation of a child inside the body of the female. Now, today will be another topic which we will discuss later on.